Hello. Welcome back to our fourth morning plenary session. Few announcements. If you won an item at silent auction, please pick it up by 4 o'clock today. So the SRC gets their money and whoever you beat doesn't just get out of luck. So please pick up your item, and then right after you pick up your item, if you pick it up at the last minute or come then anyway, at 4 o'clock we have our closing session and town hall. So great opportunity to talk about what we want to see. And then finally, if you're not doing so yet, please follow us on social media. Blue Sky, Instagram, or LinkedIn. And I'll turn it over to Jonathan Lopez.
So I should have a slide here. Do I have to advance myself is the question. There we go. Okay, this is a little short for me. So for any of you who don't know me, I'm Jonathan Lopez. I am the chair of a program that you heard maybe Amy Roseman talk a little bit about, the Headwaters Leadership Academy. And so we are relatively new to the society. We've been around for, we just finished our 4th year, but we are an early career leadership program that is aimed at developing essentially a pipeline into SFS leadership and giving back to the society. And so the folks that we're about to recognize here in a moment have completed 2 years of professional development activities, small group mentorship, and society service, right? And so if you look at the list of people who are involved in different committees across the society, you will see a lot of our representatives there. And a lot of these folks have continued to serve the society beyond their time in our program. And so first off, I want to thank everybody who responded to Amy Roseman's request for donations the other day. We really appreciate the fact that you all are willing to support programs like ours and like Emerge and InStars. So thank you very much. And then the other thing I want to do real quick before I get into calling out the individuals who are going to come up here and be recognized for completing the program is to thank all the people who have contributed in some way. So we have a pretty low budget operation that we run that is, like most of SFS, entirely volunteer based. And so I would like anybody who has over the last 4 years that we've been running this program, if you've contributed at any point, whether it's as a guest speaker or as a mentor for our program, please stand so that we can thank you very much for your time. And so you probably noticed the people who just stood up, and those are the people that we're trying to model ourselves after and give back to the society and serve all of you who are here today. So thank you very much. And without further ado, I will go ahead and recognize these people for their time and be quick so we can get on with the proceedings. But so a number of us we have 4 of us here today, and so you guys will come up here. I'll give you your certificates when I call your name, and we'll have a group photo at the end. So we'll have individual photos, and then once we wrap up, we'll get a photo, all 5 of us together. Okay, so our first person that I want to recognize is Christine Cornish. Christine can't be with us here today. I'm going to do the people that can't be here first, but we should still clap for Christine. So thank you, Christine. Denzel Cross as well. Thank you, Denzel. Kawan Fonseca. Sabrina Jaffe. Megan Malish. Miracle Oso. And now we're going to get to the people who are actually here. So Brittany Parada. Is Brittany here with us this morning? There she is. Irene Sanchez-Gonzalez. Audrey Thellman. And then last but certainly not least, Anna Vincent. Thanks, everyone.
Good morning, everybody. My name is Karen Gaines. I was the chair of the Conservation and Environmental Issues Committee. I am now the current co-chair, along with Rowan. So thank you, Rowan, for stepping up to be a leader. I'd like to present the Graduate Student Conservation Research Award. This is an award supported by our little section that is made based on the scientific merit of a graduate student research proposal that successfully addresses for conservation-related criteria. We had wonderful applicants this year. Thank you very much for sharing your research with us. And I'm very happy to award, we are very happy to award 2 research proposals this year with our award. The first is by Brady Smith of Western Colorado University. His research project is entitled Willow Effects on Essential Habitat Elements of the Rio Grande Sucker and Rio Grande Chub in North Crestone Creek, Colorado. My understanding is that Brady is not here at the meeting. Okay. And then the second award goes to Alexander Juan at UC Berkeley. For his proposal titled Modeling Consumption of Native Organisms by Invasive Sacramento Pike Minnow in the Eel River in California. Alexander, are you here? No. Well, let's give these folks the accolades they deserve, and if you know these people, their checks are in the mail. Thank you very much.
Hello, everybody. My name is Christina Lincoln. I am a co-chair of the International Coordination Committee alongside outgoing co-chair Erasme Uizai and incoming co-chair Carla Lopez Lloreda. The International Coordination Committee has the honor of presenting the Resch Rosenberg Africa-Asia Travel Award. Established in 2025 by Vince and Cheryl Resch, the award recognizes the longstanding collaboration and friendship between Dr. Vincent Resch and Dr. David Rosenberg, longtime member of SFS, former president, and one of the founders of our journal. The award supports early career freshwater scientists from Africa or Asia with travel funding and meeting registration to attend SFS. Last year, the inaugural awards were presented to Gladys Moanga from Malawi and Arundathi Ekenyake from Sri Lanka. Our 2026 award recipient is Enoro Kennedy Owenu from Nigeria. During his PhD at Rhodes University in South Africa, he developed mechanistic and trait-based framework to predict how macroinvertebrate traits influence exposure and sensitivity to microplastic contamination in African rivers. As a postdoctoral fellow at the Institute for Water Research, also at Rhodes University, he now leads projects on micro, meso, and macroplastic pollution, supervises graduate students, and collaborates internationally to develop biomonitoring tools and sustainability frameworks with the broader goal to advance evidence-based water management and contribute to global understanding of freshwater ecosystem responses to emerging contaminants. Please join me in congratulating NRO as our 2026 recipient of the Resch Rosenberg Travel Award.
Good morning, everybody. I'm Jim Hood. I'm here to do a couple things. So first, I want to talk about our fun run winners. So as you know, the 5K fun run is a long-standing tradition of SFS. This year's fun run took place on Tuesday morning, and we had almost 100 participants. So let's all give them a round of applause. So today I'd like to recognize the winners with a little memento to celebrate their success. But first I'd like to give a shout out to Shannon Clausen, who organized the fun run. She did a lot of work to put this together, and let's recognize all the work she's done. Okay, so if you're in the audience, please come up, and when I read off your name to receive your prize. First place for women under 40 was Anna French. Is Anna here? All right. First place for women in the 40 to 60 category, Kathy Jo Jankowski. Yeah, you have to stay. Just like the other winners. All right, first place for women in the 60 plus category, Amy Roseman. One big picture. All right, first place for men under 40, Justin Pomerantz. Oh, look at all this energy and athleticism. Congratulations. Thanks. First place for men in the 40 to 60 category, Charles Yakulik. Hope I said that right. All right, and then for the overall results across all age and gender categories, we'll honor the top 4, and if you already collected your prize, we'll still give you a round of applause. First place overall, Anna French. And second place overall, Justin Pomerantz. In third place overall, Noah Grode. And then in fourth place, Jonathan Gilman. All right, congratulations, everybody. Nice job. Oh, wow, I didn't see that. Thanks. That's awesome. All right, I have to enjoy that a little for a second. All right, next, I want to talk about our fundraising for Emerge, Instars, HLA, and Streams. We did that. We hit our goal of getting $10,000 for these events. So congratulations, everybody. Nice job. And so that brings us to $20,000 with the funds donated by the fellows. I'd like to encourage you, let's keep going with this momentum. These are great programs that we really want to support that our SFS members are doing. So I encourage you to take a picture of the QR code, use it, donate. To go onto the website, there's a really prominent donate button there that you can donate on. And let's keep building on this momentum. And thanks to everybody who's already donated. Yeah, like Selly's doing. Awesome, Selly. Leading by example. Okay, so my final task up here is to introduce our what? Oh, yeah. Sorry about that. There's a video celebrating SFS that I've forgotten about, which is coming.
If you think about it, our relationship to fresh water is as much a part of being human as is our own curiosity. And if you pay attention to the rivers, lakes, and wetlands around us, you might just see a community of people dedicated to understanding the nature of these ecosystems. And with good reason. These are the waters that make our planet a home, not just to us, but to a whole world of life that relies on them. As habitats, these inland waters host a vast wealth of aquatic life. From iconic animals to cryptic creatures, all interconnected in a dynamic web of life. As ecosystems, these waters produce vital resources for human life, from flood and fire resilience to nutrient processing and carbon storage, and from food fisheries to our most precious natural resource, clean, drinkable water. They sustained us throughout human history, but we now enter a future with greater uncertainty in freshwater resources, right down to the quality and availability of water itself. Now, more than ever, the world needs sound science to help model and guide our wisest decisions that will not only manage these ecosystems, but help restore those that have been damaged. Since 1953, the Society for Freshwater Science has helped organize and advance the understanding of rivers, lakes, and wetlands, and how these ecosystems respond to our human needs and footprint. Through sharing scientific discoveries, upholding the standard for aquatic research, and developing the next generation of scientists who represent communities and cultures across the globe. Because everyone needs fresh water, and we all need the ecosystems that produce it.
That's fantastic. So donate. We're awesome, as this video shows, and let's continue to build on that. All right, so my last task here is to introduce our final plenary speaker. So I'm pleased to introduce Dr. Will Clements. Dr. Clements is a professor and chair of the undergraduate major in the Department of Fish, Wildlife, and Conservation Biology at Colorado State University. Dr. Clements' long-term research focus is on the community responses to and recovery from contaminants and other anthropogenic stressors. Dr. Clements is the co-author of 2 ecotoxicology books, Community Ecotoxicology and Ecotoxicology, A Comprehensive Treatment. He has published over 100 peer-reviewed journal articles in journals such as Environmental Science and Technology, Ecological Applications, and 13 times in our society journals, Freshwater Science and the Journal of the North American Benthological Society. Dr. Clements teaches graduate and undergraduate courses in ecology, experimental design, and ecotoxicology. He has mentored 37 graduate students in postdoctoral fellowships. Dr. Clements has also been a long time and active member of SFS. He's a fellow of the Society for Freshwater Science, chaired the executive committee, and has been an associate editor for Freshwater Science since 1997. Dr. Clements is also active in the Society of Environmental Toxicology and Chemistry, where he served as on the board of directors twice and has been the president of the Rocky Mountain chapter twice as well. So please help me welcome Dr. Clements.
Wow. First off, thanks for being here. I guess I wasn't sure how many folks would show up given the last day and after the big party last night. So first off, thanks to you guys for showing up in the first place. I also want to thank Jim and Camille for 2 things, for organizing what a great session, a series of plenaries, but I guess more importantly for all the hard work they've done putting this meeting together. If you've been involved as a co-chair or a program chair or whatever throughout the course of SFS or JNABS, you know how much work that requires. And so thanks to you guys for making this happen and making such a great meeting. So I am, needless to say, honored to be doing this and a bit humbled to be doing this and a wee bit intimidated. And the last part is because I've been coming to this meeting, I'm doing the math on this, I think 84 is my very first one. And since I made that transition from studying seagrass communities to freshwater streams and lakes, one of the best decisions I think I ever made in my life, this has been my family. So it is a wee bit intimidating to be giving a talk to your family. I think what I'm going to do today, I guess, is a couple things. That the photograph you're staring at, that I've been sitting on that side and watching, why are people staring down here? Now I know that's where the big screen is. That's pretty helpful, actually. What I'm going to talk about today is a kind of a 37-year history of some work that we've done on the Arkansas and other streams in Colorado and other streams of the West, looking at the effects of contaminants. And this photograph is really, in some respects, kind of a snapshot of what I'll be talking about today, but it's also maybe in some respects a trajectory of my research program over the last 37 years, showing that, and maybe it's a function of aging, but the first half of my career really was focused on trying to understand all the nasty things that metals do to streams from abandoned mines and other stressors, other contaminants as well. But in the last decade or so, maybe 15 years, I really shifted to focusing on trying to understand how these systems recover and what speeds up or slows down that process of recovery and what these systems look like after they recover and what criteria might look like as we define what restoration success looks like. A roadmap, a quick overview of what I'll be talking about. I will show you a handful of, I feel obligated to show you a handful of some really nasty streams, but just in the background here, streams are contaminated by metals. And this is actually the North Fork of Clear Creek. It's a US EPA Superfund site. It's also, this is a tributary to Clear Creek, and it provides drinking water for a pretty significant portion of Denver's population. It's also just upstream of Coors Brewing Company, and I'll let you make what you will of that. So a couple things, I'll spend a little bit of time, kind of a general overview of mines do and the kind of a global responses and effects of mines and mining on ecosystems. Some work on factors that influence ecological recovery, a little bit of kind of my perspective on how we go about defining and perhaps how we shouldn't define restoration success. The more speculative aspect of the talk, regime shifts and novel communities and work we've been doing recently. And if those of you who are fortunate enough to be here for Angus McIntosh's plenary yesterday, there's some commonality in some of the things that we've been working on in my shop, as well as what Angus has been doing. And then I think it would be remiss if we didn't spend a little bit of time talking about climate change and how that may influence the responses to and how we characterize restoration success. You can imagine my glee when Jim asked me, I think a year ago, to do a plenary. And then a few weeks later, he asked me if I could do the entire session. So it's not going to be an hour and a half, I promise you that. And so in fact, having taught undergraduate courses for many, many years, I recognize I'm really sensitive to the fact it's really difficult to keep people's attention for that long a period of time. So I put together what I hope will be about a 45-minute talk. Okay, let's go big. This is a map, really nice study by Macklin looking at the distribution of inactive mines around the world. And it's pretty stunning. A couple of things kind of jump out at you. First off is the large majority of these are in North America. First off, it's a whole bunch of them, 160,000 or so. A really large fraction in North America, and of course quite a few of them are kind of clustered along that western edge, and there's some interesting tectonic and geological reasons as to why that is, but that tends to be in both North and South America, a large number of those mines in that part of the world. So what they do and the extent of downstream effects, both in terms of river reaches, kilometers they're impaired, and floodplain area that's impaired. Again, it is kind of a North American-centric issue, and that may be in part because we've done perhaps a better job characterizing these effects, but there's no question that these, much of the issues we see with the effects of both inactive and active mines are in North America. And to give you a sense of what this looks like, this is a really nice paper by Gilsham that looks at different aspects of global effects of mining at a fairly large scale, going from deforestation, mines in water scarce areas, floodplain effects, mining in protected areas. And I think the compelling thing about this is that depending on which particular category of stressors you're looking at, it really varies quite a bit among different regions around the world. I really want to call your attention to that lower right hand, and that should be something that is hopefully concerning to many of you in this room, the fact that a lot of this mining takes place in protected areas. And when I say that, I mean wilderness areas, national parks, not in, but adjacent to, and in some cases in national parks, national monuments, those kinds of things. And to make that case, this is a figure is put together by the National Parks Conservation Association that illustrates both the active and inactive mines that are near, adjacent to, in some cases within protected areas, wilderness areas. This should be, I think, pretty concerning to most of us in this room. And perhaps most concerning is what's happening right now up in the boundary waters. I hope many of you are probably familiar with this. It's hard to miss it. The boundary waters is, I think, one of the largest, the fourth largest wilderness area in North America. It is visited, I think it's the most visited wilderness area in the US. Was set aside, protected, has been protected since 1909. There's a great quote that I think Wallace Stegner, one of my favorite authors, he wrote this letter when the Wilderness Area Act was being debated. And he had this quote about wilderness being the geography of hope, which I think is a great way to think about wilderness areas in general. Well, when I put this talk together, I was hoping to be able to use this as an opportunity or a platform to have you, to make a plea for you to write your congressmen, write your senators. I'm afraid that ship has sailed. The House Joint Committee passed, 140 passed the Senate and became law last month. And what this did is to permanently prohibit future restrictions on mining. And before that, there actually has been in place some protections of this particular area. Permanent may be a bit strong. It just means that the next incoming administration simply can't overturn it. It will require an act of Congress, literally, to make this go away. I think it has some pretty important implications for other places, particularly in Alaska, the Pebble Mine, the Gates, the Arctic. in large part because one of the signees on this was the senator from Alaska. So I think there is a reasonably good chance we may see more of this in the near future. Okay, so a couple more slides about the potential effects and the scope and scale of this. This is another shot of that, the North Fork of Clear Creek Superfund site. And to put some dollar figures on what this has cost, the expense of this, and particularly in the western U.S., a total of 63 of EPA Superfund sites, CERCLA sites, which are, I'm sure many of you are familiar with, are the worst of the worst in terms of environmental effects and potential human health effects. In Colorado, where I've done much of my work, most of my work, 9 of the Superfund sites are abandoned mines, and that number may be moving around. There have been a couple have been taken off, and maybe another so has been added to that. That bottom figure should be pretty striking to you. That 23% of the streams in the southern Rocky Mountain ecoregion are impaired by metals, and that's not simply looking at measuring abiotic factors and metal concentrations. That's actually, I had an opportunity to work with EPA on a EMAP program when that was operational. And the way EMAP work is you sample sites randomly. So that means you can make pretty broad distinctions or pretty broad conclusions about the extent of a particular perturbation in this area. And so that 23% becomes a little less surprising when you look at the distribution, the number of mines in Colorado, but particularly in the Colorado Mineral Belt. This is a really nice slide that Travis Schmidt and his colleagues at USGS put together, characterizing both the location of abandoned mines, but the distribution of these in that part of the world, abandoned and active and inactive mines. If you look at that lower part of the mineral belt around Silverton, huge density of mines in that area near Silverton, and in particular, I think I've seen this photograph at least 6 times at this meeting here. This is something that got lots and lots of attention. As it turns out, fortunately, the visual impact was much greater than toxicological impact. Now, I'll say more about that a little bit later on. It certainly did have pretty strong visual impact in terms of what it looks like, but Colorado Parks and Wildlife actually was able to conduct caged fish experiments, I think the day after the spill took place, and was able to determine that despite what it looks like, it's mostly iron and other contaminants as well, but it's certainly the toxicological effects are much less significant than what you might imagine here. Okay, part 2, I guess, I want to spend a little bit of time talking about this great opportunity I had to collaborate with some folks from USGS, Chris Mabane in Boise, Idaho, and Michelle Hornberger and Terry Short from California on the Clark Fork work. Dave Herbst at UC Santa Barbara and myself, we all had these really nice long-term data sets, and we've been coming to NABS or SFS meetings or even CTAC meetings for many years and showing these data sets. And I think we even said, oh, sometimes we get together and let's put these data sets together and try to tell a story. We never really did that until COVID hit. So this is one of those, and you may have some of these yourself, a kind of a COVID-inspired collaboration, because no one was going to meetings at the time, so let's just do this. So we took these 4 data sets, long-term data sets from each of these 4 watersheds, and basically tried to understand various factors that result in effects of metals, but more importantly, the factors that influence the ecological recovery of that. And not surprisingly, if you know me, that focus was on aquatic insects. I'm going to show you some fish pictures in a few minutes. We did some work on fish in a different part of this study. But exclusively on aquatic insects for this particular aspect of it. And for reasons I guess I don't need to go into. Obviously, most of you probably know that there's this really well-characterized range of sensitivity among these groups of organisms that's pretty universal. You know, we don't have a lot of laws in ecology or certainly in benthic ecology, but certainly one of the things that's been really consistent is certain groups of organisms are super sensitive to metals and other contaminants, and some others are not. So it works out pretty well for a bioassessment tool. And in particular, certain groups like mayflies, Ephemeroptera, are extremely sensitive to contaminants and metals. We don't get to read about aquatic insects or mayflies specifically in the Washington Post, but this is actually a really neat article that came out a few years ago talking about the demise of insects, but more specifically the demise of mayflies and how they are such important sentinels of ecological effects and ecological recovery. And that last bit about the message being a bit grim in terms of what's happening to them. And there's been some really neat work physiological work trying to understand what's unique about mayflies to make them so sensitive. They're like little sponges. I guess the large gill plates, but certainly the permeability of these organisms, especially some of the early instars, makes them especially susceptible to a whole suite of contaminants. So here's the study design. We had, again, these 4 watersheds, the Arkansas, the Clark Fork, Leviathan Creek, and Big Deer Creek. And in each one of those, we'd been, I think probably some of the studies, some of the watersheds had half a dozen or so. I think we had probably 7 or 8. And to put these data sets together, we decided to limit it to just reference sites, impacted sites, and downstream sites. So I'll show you results. Actually, I'm going to show you results, just a couple of those, just to make the case of how these systems responded to both the effects and the remediation and recovery. The study duration ranged from 20 to 30 years, so 4 pretty nice data sets to look at that kind of before and after responses. And then the post-restoration monitoring. I think that's what makes this pretty special, is that the post-restoration monitoring, anywhere between 9 and 17 years. It's a really nice data set in order to quantify and determine what happened after you remove this particular set of stressors. Here's what the streams look like, ranging from 2nd to 4th order streams. They're all wadable, they're all cobble bottom, snowmelt driven streams, typical western streams. And that snowmelt driven part is going to turn out to be pretty important. At least a couple slides I'm going to show you, because it turns out that the annual variation in snowpack turns out to be a really big driver of some of these metal concentrations, and the seasonal characteristics as well. Because of the fact that some studies sampled both seasons, spring and summer, and some only did summer or fall, we just limited this to a single season. This is summer season, summer sampling data. And this is what the metal concentrations look like in each one of those watersheds. A couple things I want to call your attention to. The metal concentration, which is on the y-axis, is expressed in terms of the concentrations that were measured divided by what we know the toxicological concentrations, the concentrations that cause effects. So that's based on things like DOC and water hardness, a whole suite of other abiotic factors. So if you buy the argument these metals at those concentrations are additive, you can simply take that ratio and add them up, and if there's a sum of something greater than 1, you might expect to see chronic effects. And that's a fairly conservative value. So that value, that horizontal line down there, represents where we might start to expect to see chronic effects on these communities. Those arrows indicate when the remediation was completed for each one of these watersheds. And what I want, and the other important thing to point out is it's on a log scale. So those are big changes you're seeing going from left to right there. And some of these streams, like Leviathan Creek and the Arkansas River, had screaming high concentrations of metals before the remediation was initiated. But those metal concentrations came down pretty quickly as a result of the remediation that was done, and all 4 of these systems came down precipitously, and in most cases approached and stayed around that safe concentration, that safe chronic concentration throughout the remainder of the study. You see a fair amount of annual variation. Again, that snowpack, El Nino, La Nina years really do influence the amount of snow we get each year. So that certainly explains some of that annual variation you're seeing there. And what did the macroinvertebrates do? Well, this is a simple metric that I think many of us would believe is pretty useful, just quantifying the total number of species that are present in a system as a result of removing this particular stressor. Again, the same layout, the same time scale. The arrows indicate when remediation was completed. And what you're seeing, and that horizontal line is the reference. So this is expressed in terms of proportion of reference sites, so that you see very quickly these streams, at least 3 of them are just racing back up to that reference condition within about 10 or 12 years. And it's pretty remarkable. In fact, this is a mirror image of what the metal concentrations are doing. So within about 10 years or so, most of these streams have recovered. The Clark Fork took a little bit longer, and in fact, that little dip down at the end, I think Michelle and Terry were telling me as the result of some thunderstorms taking place towards the end of the record there that increased metal concentrations and reduced those macroinvertebrate communities. But nonetheless, the important thing is that all 3 of these streams, and all 4 are heading in the right direction, and most of that took place well before the remediation was completed. I think that's a pretty interesting finding of this as well. So I think one of the important things that came out of this particular study, in addition to having this long-term data set is asking questions: what determined recovery? What were the key factors that really determined how fast these systems recovered? And certainly one of the things that mattered a whole lot was the connectivity and the proximity, and this is probably not of surprise to anyone in this room, the proximity to upstream colonization. Certainly in the Arkansas and Leviathan Creek and Big Deer Creek, there's a really well-characterized, a very healthy and robust upstream reference community that flooded those downstream as the remediation took place, as metal concentrations came down. On the Clark Fork, not so much. So the opportunities for recolonization of those downstream stations were not quite as good. The other thing that turned out to be, and this is probably a bit more speculative, but it looked like flow variability. We have really good gauge data for all these watersheds, so the flow variability, and I'm going to probably go out on a limb here and speculate that flow variability may be selected for organisms that are able to recolonize pretty quickly. So those streams that had higher flow variability tend to recover faster than the other ones. The other thing I want to point out before this goes away is that each of these are continuing. We just did our 38th year of sampling on the Arkansas River about a week or so ago. I know that in some cases the torch is being passed. In some cases, instead of going to every year or twice a year, they may be every 5 years or something, but each one of these sets of data are still continuing. Now I want to spend a little bit of time, I guess the rest of the time here, talking about how we go about defining what restoration success looks like. And I think this is the California Gold Superfund site as it looked in 1993. It was designated as a Superfund site in '89. I think the Superfund came in, I think it was '86 or '84 when it first came on the books. And certainly when the bar is set so low, that looks like a bomb went off there, right? You're not going to have too much difficulty improving that, hopefully anyway. And I want to give a quick shout out. Unfortunately, there's not kind of like the other plenaries, there is no human dimensions aspect, follow up to my talk to talk about this. But I think it's really important to recognize some of the local landowners that played such a prominent role in keeping this going. First in identifying it's a pretty serious issue. Doc Smith, who was third generation rancher on this, in fact, he lived in this cabin where his great-great-grandparents built and he was born in this. Anyway, he was a local veterinarian and noticed all this really strange stuff taking place with his cattle and his horses that were grazing on some of the grass was irrigated by water that's coming in from the Arkansas River. And he and a couple of other landowners really played a really important role in both getting this designated and make sure that it was continued to be worked on for the California Gold Superfund site. So as a result of this designation, there was a restoration goal put in place early on that was designed to maintain, and this is a quote in the actual goal itself, maintain a healthy and reproducing brown trout population in the Arkansas River. I'll come back to that in a couple of minutes. But the important point about this is that in addition to a fair amount of effort to actually do the remediation, and I'm going to try to make that distinction. I've been kind of sloppy. I think a lot of us have, and maybe it's because I only learned fairly late in life that remediation and restoration are slightly different contexts in terms of what we're doing. So I'm going to try to be consistent when I talk about remediation. In this system, it's going to be focusing on improving water quality, whereas restoration is going to be mostly about habitat. And they did both of these things in this particular system. This is the other thing unique about the Arkansas River. It was the first non-litigious, if you can imagine that, a non-litigious NRDA, Natural Resource Damage Assessment. That doesn't happen too often. When these NRDAs come to fruition, the first thing to do is grab a bunch of lawyers and go into court. This one's actually able to settle it outside of court, which is, I think, a pretty remarkable feat. But the other thing about this, which is a bit unusual as well, is in that post-remediation settlement, they set aside money specifically for post-restoration assessment, to actually go in there. They put dollars in there specifically to verify, did this work? And that doesn't happen all the time. Here's what the remediation, restoration looks like. Boulder clusters, planting willows, these big rock vein structures. This is about a 10-kilometer reach of the river where they did this, these boulder cross vein structures. And this is all designed to improve habitat for brown trout, or specifically, as many of you probably know, brown trout or trout and salmons in general are kind of sit and wait predators, and they're just waiting for stuff to drift by. So a lot of this work was done to try to improve foraging locations for brown trout populations. So this is another one of those before and after remediation restoration. In this particular case, we can talk about two phases, one of which was focused exclusively on water quality, bringing those metal concentrations down. There's a period of time between about 2001 2014 in which it was not much was going on, and then 2015 and 2026, the post-restoration time period. So we have and we focus on aquatic insects, and as promised, I'll show you a little bit of work on some of the cool results of the brown trout populations, how they responded. But I want to start off first kind of putting this in context of what other sites look like, other Superfund sites look like, and in particular, what are Superfund mega sites. And I had a chance to work on an NRC committee a number of years ago in which we were given all these data sets that were from Superfund sites where they spent more than $50 million. To be a mega site, you have to spend $50 million or more to clean it up. And so these are pretty healthy expenditures to do dredging and excavation, especially if you're digging up PCBs, clearly, because you can't just dispose those when you dig them up. You got to incinerate them or some other treatment. So that's especially expensive. But these other ones, $200, $236, $138 million to do the excavation and the dredging. So these are big bucks. But what I want to call your attention to is the volume of material that was removed. And if you don't have a sense of what 2,609,000 cubic yards of stuff looks like, that's a 20-yard dump truck, which is a pretty big truck. So they're hauling away large volumes of this stuff, and that certainly accounts for the large expenditures. But I guess the point I want to make here is that, and this is the conclusion of this NRC report we did, is that even though we're spending more than 50, and in some cases close to $1 billion, we still weren't able to evaluate the effectiveness of this, because in many cases the remedy, the success was based on how much stuff you removed. It was based on, did I actually move the stuff? There really wasn't a whole lot of post-restoration or post-remediation monitoring to demonstrate there was an ecological effect or associated with this. Some other ways that we can, as I've shown you previously, other ways you can kind of demonstrate effectiveness and restoration success. I can show you pretty pictures. There's no question that those before and after photos tell a pretty good story about what metal concentrations and what habitat did in this particular system. The metal concentrations came down, the species richness, alpha diversity came up pretty quickly, there's no question. Those are really good things. So that's a really good signs, and I think most of us would agree that, yeah, that looks like that system has recovered. That little blip right there that you see, let me go back. Okay, let's just do this. So I want to make the point here is that what we're seeing with alpha diversity, yes, the total number of species are recovered, and this is something that's gotten a lot of attention recently, is that alpha diversity is a useful metric, but because of things like species replacement or homogenization of communities, in many instances we may continue to see the same number of species, but the assemblages are completely different, and that's still what we're seeing in this particular system. 20 years after remediation was complete in this system, we're still seeing these streams dominated by metal-tolerant taxa, which is a bit of a surprise. There's some really interesting theoretical literature that suggests that shouldn't be the case. That if you take these stresses away, if you take these contaminants away, that these systems should eventually return to where they were previously. And there's no question that, at least in terms of total species richness, it has. But if you look at something like community similarity and how similar composition of these communities are to the upstream reference sites, it certainly isn't quite there yet. We get maybe, if you squint a little bit, you can say 75% similarity to what the reference sites look like, but there's no question it never got back to and didn't get back to what it was at the reference condition. If you're wondering about that little blip there at the bottom, that's when they had all the heavy equipment that were moving stuff around. So we got a pretty dramatic decrease in similarity when that occurred. It also, the other thing to point out there is how quickly, after that fairly major disturbance in which it went from being about 75% similar to 40%, it came right back the next year. So these systems, again, when upstream communities are present to recolonize, really do recover pretty quickly, at least in terms of this metric. But it still never achieved what we'd expect to see or what the reference sites look like. And so this brings me to, I think, probably the most speculative, well, maybe not. I think it's one of the more speculative aspects of the study. And I think this is one of those ideas that's gotten a fair amount of attention in the restoration literature. Some of you may be familiar with some of the work that Hobbs and some of his colleagues have done. Just simplistically, you can't put Humpty Dumpty back together again. In other words, if you have some kind of disturbance that takes place and this perturbation occurs, sometimes you can't put it back the way it was previously. And I don't think you can't talk about disturbance in a group like this without showing, these ball bearing and trough and valley and hill. You have to see this every ecology textbook has this. So the idea is that sometimes it takes a fairly significant disturbance to push this to some other stable state. And in some situations, you can pick your stressor for coral reefs, and that's where most of the great work on this idea has come from at coral reef communities, because this has occurred a lot, where you have some kind of perturbation, long-term perturbations like acidification or warming, or even short-term perturbations like nutrients and boat anchors or collectors or whatever, certainly can push this to an alternative stable state. And you have this regime shift, and the compelling thing about this is that eliminating that stressor doesn't guarantee it's going to go back to where it was. And I think that's a message that Hobbs and his colleagues have certainly been propagating. It's something I guess we should be aware of and thinking about, not to overpromise what our systems are going to look like when we complete restoration. So two things I think are really germane to this, that eliminating a stressor doesn't return to where it was, and perhaps more importantly, these disturbed communities are sometimes more susceptible, more sensitive to other stressors, and that's something that I think is really important in terms of understanding what these systems are doing. So the question that we've been chasing for the last few years is asking, have these downstream communities undergone a regime shift? And we've done some experiments to try to verify, well, I'm going to show you some experiments that verify that these communities are still super tolerant to metals, but we've done some other experiments to try to understand what the mechanism behind that failure to recover is. So has it undergone a regime shift, and are these communities more sensitive to other stressors, which is especially problematic? So this is a CSU stream mesocosm facility. This is something we constructed, back in the mid-90s, I think we got this thing built. We've done all kinds of experiments in here looking at the effects of lots of different kinds of stressors. That photograph shows the mesocosms. That's my postdoc PhD student, Chris Katalik, looking at emergence, sampling emergence in these things. So that turns out to be, for different stressors, a really, really sensitive endpoint. The rate, the timing, the cumulative emergence of insects from these streams from exposure to some kind of stressor. The approach, many of you have seen this because I've talked about this work for a long time. It's pretty low key. It basically starts with a handful of rocks, and you anchor that handful of rocks in the stream for a period of time, 30 or 40 days, and then during that period of time, they're colonized by a whole suite of aquatic insects, mayflies, stoneflies, caddisflies, your favorite organisms, in pretty similar to what you see in the natural substrate. So if I took Cerber samples or Hess samples in the surrounding substrate, it's very, very similar. There's some groups that are more or less represented on these trays, but for the most part, it's a great representation of what that community looks like. So it provides a way by which we can take these natural communities and put them in mesocosms and expose them to all kinds of nasty stuff. Well, not too nasty stuff. What you're seeing there is a top view of the mesocosms and a paddle wheels driving the current there. There's a drift net that is in place. So drift also turns out to be, in fact, probably one of the more sensitive things we've looked at, just measuring the rate of drift and the drift of organisms in these things, because that is the first line of defense. I guess a lot of organisms, a lot of aquatic insects, they will, and other organisms too, they will drift downstream when they're exposed to something. And unfortunately, this is a circular system, so when they drift downstream, they just come right back to the same place. It's got to be pretty frustrating. So the design of this experiment, or 6 experiments, we've done this 6 times yet, and we're at some point, I think we're done, because I'm convinced now that maybe it's what's not going to happen. Well, let me tell you about the design first. So we've done 6 of these between 2009 and 2024. Your typical garden variety 2x2 factorial design, so 2 locations, metals, no metals from the field. Take those communities, put them in the mesocosms, and expose them to metals or no metals. So the upstream reference site, AR1, downstream, that downstream site's about 6 or 7, no, more like 10 kilometers downstream from the Superfund site. And what we're expecting to see is that over time, that greater tolerance we see in downstream communities would go away. You'd expect that. If there is some cost of tolerance, as some of the theoreticians have told us and others have speculated, that should go away. Well, it hadn't. What I'm doing here is just summarizing the effects of the means of those 6 experiments. And what it's showing, whether you look at effect size, if you want some fancy two-way analysis of variance, that's significant. So basically what it's saying is that the effects of metals on mayflies, and that was a group that certainly showed the most consistent response, was greater at the upstream, these upstream communities and downstream, and that didn't change over time. There was the most recent experiment we did had exactly the same results. So these downstream communities are still much more tolerant to metals than what the upstream communities are. Okay, why does this matter? I remember I gave a part of this talk, I think it was at Flathead Lake a number of years ago, and I said, well, is it simply academic navel gazing? And actually, we've been talking about paraphyton and biofilm, and I actually said academic navel grazing, which is probably something you don't want to think about before you had your breakfast and your coffee, but anyway. It does matter in large part, but I call your attention to this great papers written by Bob Payne of marine rocking or tidal fame. This is I think in the first issue of Ecosystems, he had this really nice paper that had reported different types of disturbance recovery trajectories. And the upper one is what you all expect. Some kind of disturbance takes place, it rushes back up and eventually reaches the same steady state where it was previously. The other two have some pretty important implications for the things we've done and some of the things you may have done. The 2 sequential disturbances may result in a steady state that's actually less than what it was previously, or in the system I'm describing now, where you have some disturbance that's taking place in a previously impaired system, which is what it looks like is happening here. And so I'm going to show you a series of experiments that we've also done looking at asking the question, do other stressors affect these communities differently? And we've done experiments with acidic pH, UV radiation. There's a time we had a couple of big projects looking at effects of UV on aquatic insects and biofilm. Abiotic stressor, stonefly predation, also showed the same kind of response. So the way this is laid out, the y-axis is the percent reduction relative to controls, The x-axis is the kind of historical exposure from reference sites to low metal sites to high metal sites. So each one of these results will be the results of a mesocosm experiment exposing to acid pH or UV radiation. But in every case, those communities that come from historically exposed sites were more sensitive to those stressors. Now, at this point, if I was a really good physiological ecologist or a geneticist, I could probably argue about what the mechanisms behind this are, but it would be speculation. We have done some genetic work on these, and much of this is a function of the communities themselves being different, the different species composition, because you're looking at, in this case, total abundance of mayflies. So have they recovered? Have benthic macroinvertebrates recovered in this system? So I guess just like there's some joke about can ecologists ever agree on something like that? Because it really depends, right? It depends on what metric you are happy with. If you think species richness is the best way to define recovery, then yeah, they certainly have. But if you're concerned about beta diversity and composition and maybe susceptibility to other stressors, perhaps not. Okay, as promised, I want to say a little bit about fish. We don't do a lot of fish work in my shop because we have some really good colleagues at Colorado Parks and Wildlife that do great work, and they've got this. About the same time we started our work on the macroinvertebrate communities, they were doing work on brown trout populations in the Arkansas, and they've got this really nice long-term data set, a bunch of streams upstream and downstream from where the metals were. And this can be a challenging system to electroshock. This is a pretty easy station, but there's some sites downstream that are a bit more robust in terms of trying to do electroshocking in this. So here's what the brown trout populations look like. This is showing biomass. Fisheries people use different weird metrics: biomass per kilogram and density per hectare. Anyway. So density and biomass, the line shows density and the histograms are showing biomass. In those 3 periods, the pre-remediation, so that difference between the first two histograms is a result of improving in water quality. So that's what happened when we reduced those metal concentrations. The second 2 bars, 2001 to 2014 compared to 2015 to 2025, is a result of habitat improvements. And so what you see there is that the density increased dramatically, density and biomass increased dramatically between those as a result of remediation, as a result of improving water quality. But habitat restoration really had no effect on density whatsoever, but the fish got a whole lot bigger. And that is really what Parks and Wildlife were hoping for. That was probably the main goal of this, is not just to improve the density of fish, but actually improve these larger fish. And so what happened in 2015, this is a really great part of the story here. In 2015, the stream was designated a gold medal trout stream, which is a big deal. We don't have many, I think it's 3 or 4 gold medal trout streams in Colorado, and this is actually a pretty big deal when this happened. However, I'm not going to answer this question because I think this is kind of infringing on that socioeconomic human dimension stuff. Was this the appropriate restoration goal for this system? Because I'm sure everyone in this room who studies fish and knows about salmonids, there's been a bunch of stuff written about how brown trout, which are not native to North America, the effects they have on other fish, particularly other salmonids, whether you're in New Zealand, talking about other native species there, in Poland, just all over the world, all over the places, these fish can certainly have a pretty devastating effect on native fisheries. So there's that, and I guess depending on who you ask, okay, well, it definitely was successful. We grew bigger fish on a lot of them, and people want that. That's a big deal. I mean, if you look at, Colorado's kind of a bit player. If you look at the national significance of sport fishing dollars that are brought in, nothing at all like Florida and Texas, but certainly $2.3 billion in economic output is a big deal for a state like Colorado, and the numbers of jobs and the number of anglers that return to this. So there is this to be considered when you define restoration success. However, there's also this. We've all experienced, those of us who live in the West, this incredible, what I call aspenification of some of our mountain towns. And the consequence of that has been pretty challenging. Certainly, obviously Leadville is a really different place back in the 1800s when the mining boom took place. But even after that, when we first started working up there, Leadville had a pretty good edge to it. It was an interesting place back in the early 80s or mid 80s. Now we have fancy coffee shops and several microbreweries. We have a clothing store that produces really high-end clothing right there in Leadville that you have to make a 6-month reservation to go in and buy 2 items. So it is, talk about gentrification, this is really happening. Okay, last bit. What about climate change? How are we going to include that? How are we going to think about that when we think about restoration success? Lots of photographs like this that you can find of what's happening in Glen Canyon Dam and some reservoir and some of the other reservoirs. And we've taken point measurements. There's a lot of slop around these lines because these are point measurements that are specific to different locations upstream and downstream over the entire course of the study. So there's a lot of variation, but there's no question that stream temperature and conductivity, this is one that kind of shocked us a little bit, have both gone up over that long-term record. So what certainly wasn't an intent of this study to demonstrate warming and increased connectivity, increased salinization, but it certainly has happened. But they're both intended and, well, not intended, expected and unexpected consequences. Some of you may have maybe seen some of this work that was presented here about the warming that's taking place in Alaska. Permafrost melts, releases sulfur dioxide, sulfur compounds, and as a result of the sulfide weathering, metals get released to concentrations that exceed criterion values. Naturally occurring stuff in the permafrost that's melting in some of these streams are actually the point that actually exceeds water quality criteria. And it may be affecting lots and lots of streams. This is, I think, maybe smart geochemists could have predicted this, but this is something that came out all of a sudden, it seems. The thing that we probably could have predicted and are predicting, this really nice study by Murphy and colleagues looking at the intersection of fires, wildfires in the West, and mining. So if you look at the distribution of mines and you superimpose on top of that what the likelihood of fires occurring, which they will, and this summer might be one of those tests of this idea, it's going to happen. And some of you hopefully had a chance to see Maricella's, or Dan Preston and I, PhD student, who's doing this really neat work, starting with the mesocosm experiments that are the factorial design, looking at crossing metals with sediments, ash from the fire that took place outside of Fort Collins, and then adding to that a field study. Again, a kind of a 2x2 design. We've got metal sites, burn sites, both sites, and reference sites. So a really nice opportunity to look at that potential intersection there. Okay, I want to finish up with I'm not going to call it a plea, but at least to make a case that I think it's really important that we demonstrate these success stories. Who is it? Who is William Randolph Hearst? The quote is, if it bleeds, it leads. And there's something about these stories that gain lots of attention. There's no question that these photographs of the Animas River after that spill back in 2015 generated lots and lots and lots of press, international press. I had friends of mine, well, from New Zealand, in fact, calling me saying, what in the world's going on in the Animas River? And again, the visual impact is pretty dramatic. So the New York Times picked this up, lots of international places picked it up. And maybe you're going to think this sounds like sour grapes, and perhaps it is to a certain extent, but our success story, reporting on the recovery of these 4 watersheds after 30-some years of monitoring, got picked up by the North 40 News. So there is this disparity, I guess, this bias that, things that generate these really images of destruction, of gloom and doom, probably generate a little more press than the success stories. And I think I guess my concern here, I'm not, my wife accused me sometimes of being a bit of a Pollyanna, and I probably am to a certain extent, but I think if we only focus on the gloom and doom aspects of what's happening, maybe the public becomes a little bit desensitized to some of the things that we're facing, and particularly when we're asking them to step up to the plate and continue contributing to fairly significant expenses for these restoration projects around the world. Okay, wow, thanks to you guys for listening. Thanks to over 100 graduate students and undergraduates who've worked on this project, or these projects. I started to put together that kind of list of all the names and what they do and where they are, and it just got to be, so I'm just going to show photographs of happy people doing happy things in happy places. They're not all smiling, because some of the conditions we work in is pretty rough, but I think the one thing that was most shocking to me, other than the fact that we've identified and sorted, counting over 2 million insects, is the fact we started this program 37 years ago with a $9,000 grant from EPA, and pretty much said, hey, Will, when I first got to CSU back in '89, Will, do you have any interest in understanding what's going on in the Arkansas River? And we've been able to continue that in large part, piecemeal funding sometimes, sometimes adequate funding, a lot of times just from the kindness of strangers helping us go out and do the collections and you can always put samples on shelves, I guess. We've done, as Dan knows, we've done a lot of that in my lab over the years. So thanks to them, and thanks to you guys again for this incredible invitation. My home university is the Colorado State Rams, and I can't make the Ram sign, the heart thing. I'm not sure I can do that. But thank you guys so much.
Hey, Will, great talk. Amazing work that you've done over the years. I have a question about your recovery and similarity. And you sort of sounded disappointment that you only saw values of about 60, maybe 70%. And you interpreted that as not full recovery, but have you compared that value to replicates at your reference site? Because I suspect they're only about 60 to 70% similar as well.
Yeah, oh, you mean like the annual similarity among replicates, or among time points?
If you took replicates at your reference site and calculated the similarity between those two replicates, I bet they'd be only about 60 or 70%.
Oh, yes. Okay, good point. Certainly individual. I mean, those are means of, let's see, probably, yeah. I mean, they're certainly means of fairly large values, large numbers of samples are collected each year. So, yeah, certainly the individual variation is certainly there. But I guess it's not so much to demonstrate. Okay, I don't mean to come across as being disappointed in that. I guess the point is that it's still pretty different, and certainly different than what the reference sites are. And in large part, I think that the fact that it's dominated by those metal-sensitive species is still pretty problematic. But yeah, I think, I don't really know at the end of the day why that what that would be like if I ran some analyses to look at certainly what the specific values would be among those replicates over that 30-some year time period.
Thank you so much. I have a question about if you had to do your experiment a 7th time, what would you choose to do it on or do differently?
So you mean doing another experiment, crossing that and looking at the continuation of tolerance? Yeah.
Yes.
It's funny you say that because I'm so anxious. Well, 2 things which some of you in the room probably don't know. I officially retired from the university a week ago, so it might be a little different to the. So I'm never really understand how to congratulations for getting old. I don't mean it that way, but it is. But I think what I probably would do, well, what I'd like to do actually more than just simply doing another experiment demonstrating the continuation of tolerance is continue some of the work that Marcella has started and maybe do a side-by-side comparison of reference streams and downstream sites to see how they might be affected by ash, for example. I think that, because that's one of the stressors we may see in that system, because it hadn't burned in a long time, that particular basin, so there's a good chance we might see that combination of fire and others. I think if I had to do that work again, I'd probably do a little more effort trying to understand mechanisms, and we have done some experiments where we've attempted to basically ask the question, is it what is about these downstream communities that's impeding recolonization? And we've played around with some of that in the mesocosms and maybe kind of chasing some of this idea that Hobbes has come up with, that the establishment of some of these metal-tolerant species is in some way inhibiting other colonists, more fragile, other organisms recolonizing. So we've done a couple of those, and it's pretty compelling. It does look and I'm not a huge believer. I've always been a skeptic of just kind of adding these ad hoc species interactions, explanations to data sets, but it does look like there's something going on there in terms of competitive capability of some of these metal-tolerant species that keep these other species from going. So I guess that's I would follow that up a little bit too.
Thank you for your talk. I was going to ask a similar question about like if you think there's competition between like disturbance-tolerant taxa in those downstream sites, but you just addressed it, so I'm going to ask another question. Has Colorado like tested the like health of these downstream sites with like an IBI or some type of like observed over expected like index?
Not ODE values, but we certainly looked at a bunch of, in fact, I've developed some, and here's an interesting, is this a criticism? Maybe it is. Okay, a lot of the work on IBI indices have been developed for aquatic insects, benthic macroinvertebrates. There are a few out there, and the problem with those is that they're usually fairly generic. It's like some groups, okay, here's a really good example. EPT, we think of EPT as being a pretty sensitive group to contaminants, but that T of the EPT is pretty tolerant to metals. So some of those, we've developed some of our own metrics because of that. And yeah, indeed, we can certainly, instead of doing the experimental work, that was probably the first motivation, was to actually look at what some of the metrics we've developed and say, yes, this is a tolerant community, this is a reference community. The concern is that there's this opportunity to be a bit tautological with those kinds of analyses, in large part because you're kind of using your benthic data to define sensitivity, and then you're using that data to come up with sensitivity indices. So there's some, I think, doing that experimentally is probably a better way to go, and we've done that a couple of times. In fact, that's probably one of the other things I'll be working on in my retirement period of time. I've got this really nice data set to look at comparing sensitivity of a bunch of aquatic insects across probably a bunch of different experiments and come up with some kind of way to, and it'll be specific to metals, I guess, not to everything else. Thanks.
Thanks, Will. Thanks for sharing that. It's over here, Travis.
Okay, thanks.
Hi, Will.
Hi, Travis.
I got a philosophical question for you. So one interpretation for like what you're saying in your talk is maybe there's processes that control biodiversity that operate at scales that are much longer than a restoration project, right? Like, so restructuring these communities might take decades or hundreds of years. And you show well, like in most of those ecosystems, water quality was actually restored. Like you can move that number, but with biodiversity being a focus of a lot of these restoration efforts. Like Clark Fork, they want 1,000 fish per mile. Like, you can't really guarantee that. So what would you say to like practitioners moving forward when they get put in a position to set goals for these restorations? Should their goal be focused on like freshwater futures and cleaning up the targets on sediment and water quality, or should their goals still maintain to be like reconstruct the biodiversity and just hope that it works out?
Yeah, it's a good question because I think, again, we don't want to overpromise the public what we can do and how successful and what success is going to look like. One thing I didn't talk about, and this is maybe it's not surprising anyone in this room, is that the habitat restoration had virtually no effect whatsoever on those on macroinvertebrate populations. They pretty much stayed the same. Just like you saw that fish, the biomass of fish, and to a certain extent the density of fish increased a little bit, but macroinvertebrates could care less. So I don't know what if I were doing this again and trying to define, okay, let me step away for a second and say, there's no question the system has recovered. There's no question that, if our goal was to restore brown trout, and that really had been the restoration goal for this project from day one. When we first started working up there, that had been, if not written in the regs, maybe actually even was in the regs when they first started, when this was designated as a Superfund site, when NRDA got involved, was to restore brown trout. As long as stakeholders agree that that's what we're trying to do, and not to say that, I'm guessing some of those stakeholders there had no interest in aquatic insects. They could care less what macroinvertebrates are doing, other than it feeds fish. I think that's a bit coarse, but I think that's pretty fair. But as long as we can demonstrate, and that's the stakeholders were involved in that decision up front, that it's brown trout we want to protect, that the things that and that, again, that's extremely successful. There's no question that brown trout populations in that stream, and I showed you those dollars for, was it $2.3 billion for the state of Colorado. The Creel surveys they've done in Leadville suggest that it added about a couple $1 million a year to the local economy. So if that's part of your goal too, then certainly that would be considered success. And we can quibble about whether brown trout are the appropriate thing to reintroduce or to maintain in a stream where they're certainly having effects on native species. But I think, I don't know whether I'd actually ever be supportive of simply focusing on fixed water quality and call it good. I think it's really important to have these other, what public or stakeholder agreed on endpoints or restoration goals should be.
I have a question.
Okay.
Actually, a couple. Do you think any of the
Thank you.
lingering effects in the invertebrate community are related to basically actual lingering metals in the system that are not necessarily reflected in water concentrations?
I think that is clearly hypothesis number 1 or 2 in terms of what's driving the failure of these systems to fully recover. And I don't know. We've done a bunch of experiments, and short-termish experiments, that we have a pretty good sense of what those metal concentrations should be and to protect those. We haven't done a full life cycle tests over years with aquatic insects. So there's no question that those metal concentrations, while they're hovering around 1 or so, they bounce around. That's the other issue too, is that I didn't spend a lot of time talking about the seasonality and the annual variation, that those peaks and valleys, those peaks actually might be having detrimental effects on these communities. So I'm not willing to simply dismiss the metal concentrations, but certainly the difference between upstream and downstream is pretty minimal for most, especially now, especially the last few years. So I'm kind of on the fence there. As an ecologist, I really want to believe that there's some ecological things going on there, but there's no question that those metal concentrations are still slightly higher than what we call safe. But I still believe that cumulative additive reflection of simply adding up all those potential concentrations that are slightly below 1 and you get 1, that's pretty conservative. That value is pretty conservative.
Okay.
Hi, I'm Chris Medlin and got to ride along on that last project. And I was going to say on this last point that I can't prove it, but it's my belief that it's both. It's both the biological stability. Once the neighborhood flipped over and then new kids started showing up, the old ones didn't just leave. It's like we like it here and we're not leaving. But the notion that an aquatic life criteria represents a no effect level is not accurate. And so even when they're added together, so my part of that, I had the simple site. There were really only 2 metals in the Big Deer, Idaho one, copper and cobalt. And so it was well below the cumulative criteria unit where we just add them together. But it's my belief that there are still lingering metals effects. And to some extent, then working with Travis Schmidt, who learned about mesocosms from Will, we did do a mesocosm type experiment with copper and cobalt, which also supports that you do, the aquatic life criteria are great. They restore, protect most things, but not everything. And mayflies sometimes are not protected.
Thanks for your talk, Will. I wanted to ask kind of about the cultural shift that you're talking about, this aspenification, and if you think that has to do with how folks in restoration are choosing their goals, like if that has to do with why brown trout were the restoration goal and how we could potentially shift the viewpoint in Colorado. I work in mine-affected fens in Silverton. Like, how do we get people to care about fishless systems and still want to do that restoration, considering the economics are probably coming from folks that may not full-time live in the state?
Yeah, I mean, there's no question that not everyone in Colorado embraces brown trout or embraces fishing in general. So I think that's quite true. When you look at, Colorado Parks and Wildlife, for example, which is the agency that sells fishing licenses and hunting licenses, a big part of their bottom line is exactly that. But by the same token, they certainly benefit from having opportunities for people to go out and observe wildlife and take the binoculars out. I don't know when they put this restoration goal for brown trout together, however many years ago, no one was really thinking about Leadville becoming aspenified, because it was a pretty rough place back in the day. So I guess I don't really have an idea how we could actually, well, there have to be some things that are put in place outside of brown trout or outside of these aquatic ecosystems to make people appreciate it. And I think the little short clip we saw a few minutes ago is a really good starting place for that. I mean, you can't watch that little short video that Judy Lee and Jeremy put together and not just feel something really special about that. So maybe that's where it starts. Maybe, appreciation for, people love to fish, people love to hunt, people love to, spend time for those recreational reasons. But certainly that part of what we represent, what the society represents, I think that's a really important place for us to demonstrate that it's not all about catching big fish or shooting big elk or whatever.
Thank you. Is it on? Okay. I'm curious about your tolerant taxa and whether some of them are species in the same genus as the ones that are intolerant, and that you may have competition or other interactions between closely related organisms that are preventing the intolerant ones from coming back.
Yeah, the experiments that I alluded to, when Jim told me I had a full hour and a half and I had to pare some stuff down, when he told me I had 40, no, I'm just kidding. I was going to spend some time talking about that set of experiments, but the species that are tolerant, that appear to be having the greatest effect on these upstream species, especially some of the mayflies, are going to be some of these case caddisflies that do a pretty good job of keeping everything. And I've seen there's a little bit of evidence out there that certain species of caddisflies can outcompete some of these other species. That's probably part of it, just the difference in the composition. It very well be, and we started when I had a PhD student a number of years ago. This is back when we're doing genetics looking at allozymes, so pretty crude techniques. There are definitely some pretty significant differences within the species of Baetis bicaudatus and Baetis tricaudatus, population bottlenecks. There's certainly a genetic component, and we we've tried hard to relate those to specific mechanisms that might make those organisms tolerant. So, and we didn't do a very good job of that. I don't think, in fact, it didn't really show up. There may be some of that going on, but I think it is species replacement and the fact that there are some of these species downstream that are in, and some of these caddisflies are at huge densities downstream. So you see entire rocks covered by Brachycentrus and huge rocks covered by Arctopsyche and some of these large caddisflies. So I think that is probably the main thing. And from my little mesocosm experiments, we've done a couple of these. That seems to be the case, that those competition perhaps played a role in preventing these other species from recolonizing these downstream trays.
First, Will, I want to thank you. I think many of us have been beneficiaries of your generosity in helping us work in our own systems. So I just want to express my gratitude for all the resources you've given us and our group to conduct experiments in the Central Appalachians. One thing that we find in the apps is in the fall, the conductivity is much higher and we have much lower stream condition based on the community composition. Do you see the same thing in your systems, and do you think that's going to be exacerbated through climate change, just with projections of how the influence of groundwater versus precipitation is going to influence ion concentrations?
Yeah, that conductivity increase that again, one of the things that we weren't expecting, we did see over that record, was pretty surprising, and it's certainly approaching those levels we might expect to see potential effects. There was a bit of seasonality to that as well. I don't think we've seen the same thing you're describing in our systems in terms of how conductivity changes between the seasonally, but yeah, there's no question that those conductivity values superimposed on slightly warmer temperatures and maybe some residual metal concentrations, as we just discussed, certainly have the potential to exacerbate some of the effects we're seeing. And there have to be some other things, other issues that are associated with the climate change, was the hydrologic changes. I mean, this is going to be a really good opportunity when we were up there sampling 2 weeks ago, I mean, I've never seen the stream that low before, so there's no question this is going to be maybe a really good test year for lots of things in the West to see how this plays out. But we haven't, despite the fact I've done a bunch of work on conductivity and the effects of that in some of the Appalachian, well, at least mostly modeling things, but we haven't really paid much attention to that in these streams that are pretty far away from any source of elevated salts. So it's pretty surprising we saw that.
Well, thank you so much. Do you have one more question? Okay, go ahead.
Hello. Great talk. I really loved all of the community tolerance you were talking about. And with the return of brown trout, I was curious if you guys have looked into it all, if the change towards a more tolerant insect community is potentially a more preferable diet for the brown trout.
You are spot on, because I mean, if anyone ever wants to come to Colorado and fish that gold medal trout stream, you want to fish with caddisflies. They're thick. That is what that river is known for. So that shift that we see from upstream, I mean, in crude forms, it's lots and lots of mayflies and some other small things, but the downstream below that, where that mining was, these caddisflies have been established, and brown trout love them. That is the preferred diet. We've done a bunch of diet studies, continue to do feeding habit studies on these brown trout populations, so it's an interesting dichotomy, because if you look at the prey availability and the calories per insect upstream, it's much, much, much less. These big hydrocyclid caddisflies and some of the brachycentric caddisflies these brown trout love these things downstream. And I'm sure Chris Katalik did some bioenergetic work on these in one of the papers we published a few years ago, and there's no question that these caddisflies contribute significantly to the diet, to the caloric, to growing these big brown trout. So there's that connection too. It's really important.
Super interesting. Thank you.
Okay, thanks so much, Will. Let's give Will one more round of applause. And before you take off, I need to make one important announcement. So if you have the time to stay, please do. I just want to address an issue that happened yesterday and make sure that we're prepared to deal with a future issue like this. Our BIPOC group met in the evening for a discussion, a productive discussion with many members. They met here in the venue. They were kicked out unceremoniously by the security, and ASN and the leadership were not here because we were getting ready at the other venue for the social. And they really didn't know what to do. They didn't have a contact. So they went out into the greenway and they sat and they met and they were harassed by local community members. It was unacceptable. They were scared and they didn't know who to contact, but they didn't feel like it rose to the level of a 911 call. That should never happen again. So Aileen Losey, thank you, our ASN coordinator. There's a phone number here that everyone needs to write down and share. And if there's anything like that that happens again, call the number because we are here to support you, and it is not acceptable behavior. Thank you. Have a good day.